Hello everyone, we are from Group 2 of the Advanced Biochemical Engineering Project. Our topic is about inactivated vaccine for COVID-19. Allow me to introduce our team members. These names are listed in order of presentation. We have Marcelino, Krisna, Dewantara, Chuanyin Ju, Zhuoying Deng, Kefei Zheng, and Hanjie Xu. Okay, let's go to the first slide. First, we talk about a brief history of vaccine. As you can see in figure 1, the timeline of the development of the vaccine. The first vaccine was discovered in 18th century, or in more specifically in 1796, by Edward Jenner and intended to confer immunity towards smallpox virus. The word vaccine itself was derived from Latin word vacca, which means a cow. Next, we can see in the 19th and 20th century, there were major advances in vaccine development, including Louis Pasteur rabies vaccine, tetanus, diphtheria in 1880s and early 1990s. Also in 1930s, there were major advances in lab techniques, allowed the cultivation of virus on the membranes of chicks embryos, which led to the development of influenza and yellow fever vaccines. The evolution of cell culture then led to the creation of polio vaccine and also marked as the beginning of the golden age of vaccine. During this period, several important vaccines like the measles, mumps, rubella, and varicella vaccines were developed. The introduction of recombinant DNA and whole genome sequencing emerged as groundbreaking innovations in the vaccine development. Now we talk about vaccine for COVID-19. As we know, there was COVID-19 pandemic caused by SARS-CoV-2. As a result of this pandemic, many countries are concerned about this pandemic, as we can see in figure 2. There is emergency news about location and statement from government. Also, as we can see, there is a news about how innovation is helping and so on. So many countries continue to battle with the development of COVID-19 vaccine. The development of COVID-19 vaccine itself following several steps, as we can see in figure 3. In figure 3 is the common steps of vaccine development and duration of each steps. There are preclinical, clinical, which contains of 3 phases, phase 1, phase 2, and phase 3. And after that, there is monitoring. The duration it takes is approximately 3 years, but for COVID-19, the duration much shorter, 6 to 9 months. From basic science to phase 1, it takes 6 to 9 months also. From phase 2 to phase 3. By April 2020, there are 115 vaccine candidates, and from World Health Organizations in 2023, there are 183 in clinical trials and 199 in preclinical stages. Also from figure 3 shows the vaccine category and the development phase. This is the data from paper 2020 by Wang et al. As we can see also, there is a category called inactivated vaccines. Inactivated vaccines is vaccine types which kill the virus to stimulate the immune response without causing disease. It is known for the production of influenza virus before the COVID-19. It has 2 major advantages: stable storage and proven production.
So the active ingredient in the inactivated vaccine is antigen protein. It is the protein on the surface of virus. Like in the COVID-19 virus, we have genomic RNA. It is bioactive, so we don't want that. And we use some sterilization to kill them. But we keep the structure of the virus because the protein on the surface is exactly what we want. So when the vaccine is injected, this protein can react with our immune cells, and our immune cells can have a memory of this kind of protein. And our immune system can get to know how to deal with that kind of protein. So when the virus actually comes after that, compared to those who are not injected, our immune system can have a stronger response to that virus and can clear them on the spot. So to produce this kind of vaccine, we need enough protein. Enough protein means enough virus. So to grow the virus is definitely a main step. But we know virus just grow in the cells. So the first step is to grow the viral cells. And after we got enough cells, we just grow the virus in the viral cells. And now we got enough virus. But again, virus is not something we want. We want the protein. So the next main step is to sterilize. We kill the genomic RNA and we get a virus surface and it is not bioactive, so that we can inject without really being infected. After sterilization, some vaccines also add a kind of absorber to boost the immune response, but it's optional. So whether you absorb it or not, now the vaccine can be kept into proper condition and it is ready to be injected.
The virus cultivation process of COVID-19 inactivated vaccine can be divided into 3 steps, each of which is closely linked. First, researchers need to isolate the original COVID-19 strain from clinical samples, confirm its genetic characteristics such as S protein sequence through gene sequencing, and select viral cells as host. Viral cells were selected because they are highly sensitive to COVID-19 virus and suitable for industrial culture. Subsequently, researchers will place viral cells in a bioreactor, such as a basket bioreactor, for expansion and use a culture medium containing fetal bovine serum to promote cell adhesion growth. When the density of viral cells reaches 18% to 19%, researchers will start to inoculate COVID-19 virus, and they let the virus absorb the cells for 1 hour and then change the fresh medium to avoid excessive damage to the cells.
After 48 to 72 hours of culturing the virus inside cells, researchers will observe the cytopathic effect under a microscope to confirm whether the virus has successfully proliferated. And finally, they will remove cell debris through techniques such as centrifugation and filtration to harvest high-concentration virus solutions.
One of the key steps in vaccine production is sterilization, which is the inactivation of the virus. The core goal of these steps is to completely eliminate the infectivity and proliferation of the virus while retaining the integrity of its key antigenic structure, such as the spike protein, as much as possible. Early inactivation methods mainly relied on heat inactivation, but high temperature can easily cause antigenic protein denaturation or structural damage, so everything is screwed. Another attempt was ultrasonic inactivation, but since songs will have difficulty in go through protein micromolecules, the inactivation effect is limited and also cannot meet the needs. Therefore, a more reliable chemical inactivation process using beta-propyl electron (BPL) has become mainstream. BPL directly acts on virus RNA through an alkylation reaction, effectively blocking its replication ability. Compared with other inactivators such as formaldehyde and DMSO, BPL has significant advantages such as minimum conformational damage to virus surface antigens, which can retain the natural structure of antigens to the greatest extent. After inactivation process, BPL will be hydrolyzed into products with no toxic side effects, and no additional steps are required to remove residues, thus simplifying the production process. The technological improvement not only improves the safety of inactivated vaccines, but also provides a reliable means for rapid response to infectious disease.
As my teammate introduced, inactive vaccine has been widely used. However, what about others? There are quite a number of vaccines here. In fact, the most widely used vaccines are inactivated vaccines, mRNA vaccines, viral vector vaccines, and recombinant protein vaccines. mRNA vaccines consist of mRNA fragments encoding viral antigenic proteins encapsulated in lipid nanoparticles. Their benefits include rapid development and production, as well as strong immune response. However, they have strict cold chain requirements, are less stable, and are costly to produce. Viral vector vaccines use harmless viruses to carry viral antigen genes. While they can stimulate comprehensive immune response, their effectiveness may be reduced by pre-existing immunity to the carrier virus, and they carry potential risks like GMO integration. Recombinant protein vaccines are made by producing viral antigens protein by genetic engineering, like the spike proteins. They are safe and have low production costs, but their weaker immunity often requires adjuvant. Inactivated vaccines generated via chemical or physical inactivation of virus. They are highly safe, especially for individuals with weak immune system, with mature and controllable production process. They are relatively easy to develop and produce. They can stimulate strong and broad immune responses and are thermally stable, make them easy to transport and store. However, they usually require multiple doses, and their effectiveness can be significantly affected by viral mutations. To sum up, inactivated vaccines, with their advantages of safety, stability, and minimal cold chain requirements, can be easily distributed and used in various regions, particularly those with limited medical resources or underdeveloped cold chain facilities. As a result, they have become the most widely used vaccines to date and are instrumental in achieving herd immunity. Some of the data and images used in this video may be from public materials or papers. They are only used for educational purpose. We respect copyright law and intellectual property rights. If you have any copyright issues regarding the materials presented in this video, please contact us promptly and we will take appropriate actions.